Thanks so much for the introduction, Ahmed. Really excited about the session today. So as all you know, there's been a whole lot of discussion recently in the space about not just potential for scaling of cellular agriculture, but also methods and tools to start thinking about how we can address these challenges. So as the field continues to expand and we continue to pull more aspects of standard biotechnology into the cell-based meat space, it's really exciting to start to see what a variety of solutions, options, and possibilities we have in the area. So basically, I think that's really going to be the heart today of our conversation is more than anything, what can we bring to the field of scaling that is new, that people have not thought about, and what can take us to the next level in cellular agriculture to be able to start thinking about these questions in a deeper way. And with that, I'm going to hand the floor off to Jacqui Matthews, our panelist from Roslin Technology, who's going to give a short introduction on herself and her background, and we'll go from there.
Okay, thanks very much, Kate. Lovely to meet you all this evening, this afternoon, this morning, wherever you're based in the world. So I'm Professor Jacqui Matthews. I'm the Chief Scientific Officer at Roslin Technologies. I originally trained as a veterinarian and spent nearly 30 years in academia leading animal science projects targeted mostly at vaccines and diagnostics development. But I very much worked in the innovation space. I led over 40 projects valued at over £13 million in funding and had a lot of outputs from that, publications, 140 publications, but importantly, policy outputs and impactful outputs such as patents and a commercialized test. And working in that space encouraged me to move over to Roslin Technologies in 2019 as their CSO to develop and shape their R&D program. And over the last 2 years, we've shaped our program to become world leaders in livestock and other domestic animal-induced pluripotent stem cells. And that's what we bring to the cultivated meat sector. And I'll tell you a bit more about those later. Back to you, Kate.
Wonderful. Thanks so much, Jacqui. We're really excited having you join the conversation. Alex, could you give a little bit of background about yourself as well?
Hi, I'm Alex Buko. I'm Vice President of Human Metabolome Technologies here in Boston. We're a metabolomics company. My history actually has been over 32 years in pharma and biotechnology, leading analytical groups. And part of that support was cell line development for pharma and biotech. In HMT, and I've been here about 8 years, and so it's a little bit, almost 40 years of doing metabolomics and proteomics support. We've been really interested in following cellular agriculture for the past 2-3 years now. And what HMT can do is we can actually measure the metabolites in cell media, cell tissue, in your early product development, and to look at these things on the micro scale, so microliters of cell media. We can look at a million cells, or we can look at a few milligrams of tissue and give a readout in terms of essential fatty acids, essential amino acids, vitamins, all those type of things that can help you develop the cell line differentiation, or your end product, end product as well. So that's
wonderful. Great. Thanks so much, Alex. Kerem, could you give a brief introduction as well?
Thank you, Kate. Nice to have you here. And I'm actually non-PhD guy in this session, I believe. I'm an electrical engineer and computer scientist, a serial entrepreneur. I used to work for a corporate for 8 years, then moved to clean tech space. I started with my agriculture data generation startup, then climate adaptation startup. And this is my third endeavor with 4 founders. There are 2 scientists in our team and 2 businessmen, and I represent the business side of our company. We are developing animal-free, non-GMO growth medium supplement in order to make cultivated meat more affordable. So we have some novel ideas in that area, and we utilize our scientist experiences and bring this product for cultivated meat companies and startups to achieve more sustainable world. Thank you.
Great. Wonderful. Thank you so much. So At the heart, we've got a lot of people who are working on bringing some cell line technology that may not have been traditionally present in this space as a service provider into the space. So Jacqui, could you elaborate a little bit on some of the cell line development that Roslin Tech is doing for the cellular agriculture space?
Yes, thanks, Kate. So we have, well, we've got two types of cells that we have available for the cell ag space, induced pluripotent stem cells and also primordial germ cells. We mostly focus on IPS cells. We already have lines available. We have commercial contracts and evaluation licenses in place for pig IPS cell lines. And the advantages of these lines is that they have an absolutely tremendous self-renewing capacity, and that will address one of the key scale-up challenges, because you don't have to keep going back to the live animal to derive new cell lines. We're already at 400 doublings and counting with our pig IPS cell lines. And they have a doubling time of about 16 to 24 hours, which is pretty tight actually. And we're at 60 passages with those cells. So we believe these are the type of cells that the cell ag sector will really need to scale massively going forward to reach their challenges of cost, price parity, and sustainability. So we offer these cells already today for people to try out and test in their systems, and we work with partners either using their media and with our own media too. And we're already, even with those lines, addressing the cost of that media to reduce the price of the cell culture and the cell scale-up picture. It's worth noting that our media is fetal bovine serum free, so that is also a big challenge of the industry that we've already addressed with these lines.
Wonderful, great. And I know one thing that was really exciting about our conversation yesterday as a group was just how much crossover there can be too when you have that cell line development, what you're able to do with those cells and that media as it develops, and what kind of biomarkers and different types of metabolomic analysis you can bring to get more insights on cell type, cell scaling, some of the considerations around flavor. Alex, could you maybe elaborate a little bit on what you're able to do once you have a cell line growth media inputs potentially from a client who's interested in working with you?
Sure. I mean, in drug development, we always had that the issue when you scale up, your profile can change. And so whether it's a small molecule or a biologic, when you go from small fermenters to large, you scale up to clinical trial size, that the substance can change, but also the environment, the contamination, everything around it can change. So I surmise in the same conditions here, when you have a small pilot and you generate a product, pre-product, early product, you have a certain profile. But as you scale up and try to mobilize a larger product development pipeline, you still want to keep tight control over that, those cells in terms of the profiles, the metabolomics, profile of that product. So what we measure then in cell media, we can measure essential fatty acids, essential omega-3, omega-6, omega-9. We can look at various flavor components, osmolytes, stress components, oxidative stress components, things that can give a negative feedback to hold both growth, but also those that are there to help promote growth. So growth factors and et cetera. So, we could start at the cell lines, we can look at cell media, we can look at both, we can look at tissue as well, tissue pellets. So all these things give insights in terms of the composition of your product, but also the cell media that's surrounding it.
Great. Yeah, so for someone who's not a scientist and who's interested in kind of understanding what a group like yours can do for the field. So say you have a product that comes in the door and someone's interested in, okay, what does this taste like? How can we make it taste better? What kind of things would you ask them about?
Well, the various amino acids and other types of molecules that one associates with taste, but the taste is still a complex solution to monitor. You look for usually what we're looking for is different development products, and then compare the composition of those products. So you look at the elements that are more abundant glutamic acid in one, some other type of molecule and another, those that have been associated with taste, and you look at that different profiles and how they compare, sour, sweet, and so forth. And so that's what we would normally do. So if you just look at one product, it's hard to say this is going to taste like something or other, right? But we're looking for those in different, as you change the cell media, the changing conditions, how does that profile change over time? And then that would give you some idea of which direction to go to if taste is something you're trying to optimize here.
Great, that's really helpful. And so say you have a group that comes in the door, they're really interested in, they just say they want a better taste, they want better cell growth. What are the first go-tos that you tend to look for or think about?
Well, usually looking for nutrient flux, so are you running short of essential amino acids, running short on antioxidants and vitamins? So so do you have plentiful supply around the cell? If the cells have a high nutrient flux, you're depleting some of these essentials, then that's going to work against you. Or look for various toxins that might also be, again, a certain amount of toxins are normal and expected, but if you have a high level of putrescine or some other that might give you a negative feedback as well. Taste, again, is harder to measure. What you usually do is we're just kind of partitioning up the more abundant metabolites that might be associated with taste and just look at how they differ versus the different production lines that you're looking at.
Great, that's really interesting. And so Kerem, I know you've got some really interesting solutions on your side. You're working at it, Biftek, to work around some of these challenges to look at different inputs, especially with cell growth media. Could you give us some color to some of the areas that you're investigating that could revolutionize these technologies?
Yeah, thank you. Actually, there are some known methodologies like playing with the genes of host organisms like barley, some insects, and obtaining desired molecules out of that by recombinant growth factor technology. So we are moving from a different path. Actually, what our scientists bring on the table is that we are living with microorganisms in our guts, right, as a mammalian. So actually, we have 2 kilograms of microorganisms in our guts, and those microorganisms are very beneficial for our mental health, immune system, as well as muscle growth. So we picked this muscle growth idea from there, and we screen lots of microbes from different sources like bees or dolphins or cows, and we found out that there are some very beneficial microorganisms which is good for cell proliferation and cell growth. So we are using the power of nature. We are using some natural ingredients and use this power for the meat applications. Nowadays, we applied for aerobic bacteria, for IP, a patent application, and now looking to anaerobic bacteria in that area. And I believe we have some very promising solutions. And like our company, there might be some other approaches to the problem. As the number of approaches grows, I believe this ecosystem will make culture meat more affordable. So everybody in the ecosystem is trying to do it commercial, right? If it becomes commercial, then we can proliferate as well. And other startups can interact each other. And we are very happy to share our supplements with other cultured meat startups around the globe. And I think in a couple of months' time, we will distribute our first samples and we will receive some feedbacks to make it more optimized, more excelled. So this is our aim.
Great. Yeah, wonderful. And so when people work with you, are they looking to kind of reach out and, for instance, share with you information about their cell lines and their technologies, or are you looking to really do an off-the-shelf kind of solution for companies and others who maybe just want to look to kind of supercharge their current operations?
Yeah, actually, now we specialize in red meat, the cow meat, actually, the cattle, and then we will move to fish and some other products like chicken. Um, in the upcoming months, we are going to produce our own meat by use of bioreactor, so that we can introduce the technology as it is. So if you can use our supplement and our media, then you can end up with the proliferation and cell growth. So this is going to be the stuff. Or in the meantime, for the next 6 months, I believe, we will send our supplements, and we will see if this supplement is going to work with the already established manufacturing lines in the startups. So I don't know the exact answer. I believe our solution should be customized for each different cell lines. But for each species, our product may be a standard solution. Yeah.
Great, that's really helpful context. So Jacqui, say someone comes to you and they're looking to think a little bit harder about what maybe goes into their cell growth media. Maybe they've talked with people like Kerem, maybe they're working on developing a line, maybe they're looking for one of your lines. What are those kind of top characteristics that you tend to think about or that you'd ask them about in some of those early conversations?
About scaling up for their media?
Yes.
Yeah, so I mean, we'd initially ask, obviously the key attributes that they want for their media and match it to what we've already got. But we also would be looking and ask them at what scale they're working at. And then, we would offer them the option to do a cross comparison study for cell performance. We would be cognizant of the various challenges that we have ahead of us, including costs, so where they're sourcing their media from and the level. I see someone's already asked about, the quality of the ingredients, whether it's food grade quality or pharmaceutical grade quality, what their aspirations around that would be. So far, we've worked in pharmaceutical grade quality because obviously we're deriving and establishing the cell lines. So we're doing that, to high laboratory standards. We would ask them where they would, plan to go in future, the types of cell lines, whether they would want to scale and then differentiate themselves, or whether they would want to utilize some of our technologies that we're developing. We've already shown that our cells can differentiate to both pig cells to both fat and muscle at high efficiency, and we would be asking them what their media needs would be, whether it would be to scale up their iPS cells and in addition to look at in-process differentiation methodologies. And we would tailor that for the type of company that and obviously the resources that they already have, whether they have their own lines and they want to do a comparison. And I suppose we would look at the they'd probably be keen on interbatch consistency, and that's something that would be a key attribute of the iPS cell lines because they just come from a single derived source, and we would look at that as well in terms of comparison.
Yeah, wonderful. So Alex, Jacqui's touching on some key points about working and making sure things are at, say, for instance, pharma grade. When you think a lot about, say, pharma grade products and what you'd like to see cross over and keep consistent between cell egg products and pharma products, I'd love to hear your thoughts around that. Also, things that you just see as key differences as the field continues to grow and scale.
Yeah, I mean, in this area, there are some things that are unique. For example, amino acids. D versus L amino acids. They taste different. Some are bitter, some are sweet. And some products like chicken have a higher percentage of D amino acids. So that's something that's unique to this type of product line that normally that we wouldn't be, for metabolomic studies, we don't pay too much attention to, though it is significant in various cancer areas. But the thing that came to my mind are biosimilars. In pharma, of course, we have biosimilars come up. We have patent protection, and the biosimilars come up for generics. And there's a high amount of correlation study that has to be done to show that the biosimilar has a similar efficiency and safety profile as the innovative drug. And the same thing here, you're creating a product that where you want to mimic the texture, you want to mimic the taste for market acceptability, right? So you're not just creating a powder or tube or paste that the restaurants might use. You want something for the consumer to approve of and to use. And so we want to try out to have, so one of the things we would do or like to do is compare the profile of the chicken, natural meat, natural chicken, fish, and so forth. Compare that to the profile of your product, and how do they match up? We can't tell you texture, but we can tell you something about the nutrient composition. We can tell you something about the taste values and things like that. That at least you can see in a similar way we do with biosimilars, where we examine the biomarkers or the structure and the composition of a drug product, an innovative product, to that of the biosimilar. And so the same thing would be, I would see that we would do here.
Great. Yeah, that's really good context. So for those of you who maybe haven't taken organic chemistry class in a while, what Alex is talking about when it comes to L and D amino acids is their handedness. So not only just what those amino acids are, but exactly what they look like and how they work. So if you want to learn more about that, I heavily encourage you Googling racemates and some of that work, just because it's interesting to think about a lot of these factors that I think most of us just might overlook in your day-to-day consideration of kind of some of these cell growth media components, and why it's so interesting and useful to be able to talk to experts like Alex and others who really, I mean, a lot of these experts that our go-tos are really technical contexts where these are kind of central considerations every day. So really
taste is important for these products. The taste receptors are functional lock and key, right? So you have to have the right structure fits in the right key, right receptor to have a sense of taste of whether it's sweet or sour or umami, whatever it happens to be, right? So, it makes sense that the specificity of these metabolites are important in terms of taste.
Yeah, it's extremely important to kind of think of those, and that's such a small level consideration, right? It's like much smaller than even thinking about. So most smells are small molecules, but even smaller than that are amino acids. So just thinking about these micro details that impact what a food could be like, and the level of molecular control we could have. So people like Alex and Jacqui can start to think about what we could do with food. So Alex, what's an example, if you have any? If you don't, that's completely fine too, of a food that we couldn't really get in nature that you could think about making with kind of some of these custom additives or manipulations that you've been describing?
I'm not sure what would be novel. I mean, our clients are dealing with fish, they're dealing with meat products, and chicken products, the normal things you would imagine. In an off-related field, I have also people working on algae, plant as well, in the terms of high production greenhouse, and the different nutrient elements of those products could bring to the table as well.
Great. Yeah, and Kerem, when you're thinking about additives to your media mixtures, what kind of thought process goes into that? What components of this process are you thinking about adding that you might not be able to get through different means?
Yeah, actually, the taste is very important stuff. The texture, the feel, the smell. So there are different ways of doing it, I believe. Actually, those kind of stuff was made by chefs, right? By trial and errors in the kitchens. But nowadays you can find some libraries, and those libraries can introduce the aspects of the ingredients. For instance, there are some startups I know in the United States that can differentiate different kinds of plants by some structures and some predefined parameters. This can be done for the other types of ingredient as well, I believe. In our case, we are trying to create a library of microorganisms. So those microorganisms are beneficial for instance, cell growth sometimes. Sometimes they halt the contamination. Sometimes they are used as toothpaste, which is beneficial for our teeth. So every kind of microorganisms may introduce some different stuff like amino acids or any other stuff, and that may change the taste, structure, the feeling whatsoever. Nowadays, we are not aware of those facts, but we are investigating. Every organism has some different roles, and those roles can define the taste and some other important parameters.
Great, wonderful. Thank you so much.
Actually, I had a question for Jacqui. So you had you had a question about what other sources of cells, and dawned on me, I used to like ostrich meat a lot, and it tastes like beef, looks like beef, and they said it was leaner and healthier. So the question is, when I think of these meat products, I'm always thinking of cow. But have we looked at all the types of game that traditionally on the table was perceived as a healthier meat, leaner meat?
Absolutely. There are various companies around the world that are doing more specialist material. There's one company doing cultured foie gras, for example, to stop poultry and geese being force fed. We've currently got pig cells, and we're stretching out. We've got lamb cells in the lab, which could be seen as a fairly healthy meat, and bovine cells in development. And actually, I guess the world's your oyster, to excuse the pun, if you were wanting to derive, if you had the technology, companies like ours are diving really deep to try and develop the technology to produce and generate self-renewing cells. So you could go to any host tissue, theoretically, and generate either more healthy meat. I think the option as well for cell-cultivated meat is, as you know, you can very precisely decide what is going to be in your end product, and you can gauge that very specifically with the amount of fat cells and muscle cells and other components that you would put into the end product. So you could actually program your system to produce meat or fish product at a specific level of nutrition and fat component capabilities.
Yeah.
I think there's one company in Australia that's trying to work in the space of these. I think they're doing kangaroo meat, for example. So I think the opportunities and options are endless.
Well, that's what we've been looking at. We've been looking at the ratio of saturated versus unsaturated fats, omega-3, omega-6, omega-9 ratios in different product development. So if you have ostrich meat because it's rich in omega-6, you can dial that into your product development using beef or cow or whatever bovine cells, as you say. Yeah.
Very exciting. Yeah, and that's just the tip of the iceberg, right? Is all these things, people like Alex and Jacqui have this molecular level control that they can start thinking about when it comes to these cells, as does Kerem. So it's very interesting to start thinking how you can play with these. So I'm going to hop real quickly to any questions from the audience. So if you all are interested in asking particular questions, please do jump into the Q&A or into chat, either works. And we're going to see how many of these we can cover, because it sounds like you all have been quite engaged in the conversation. So starting at the bottom first, Jacqui, is your cell culture media animal free or only FBS free?
So at the current time, it's FBS free, and we use an FBS replacement. Some of the components are derived, some of the small components are derived from animal sources, and we're working through revising our media components to make them entirely animal free. So watch this space for that. So we're on the steps to achieving that mission.
Very exciting. And Kerem, since you're working on media additives as well, are your additives animal free or FBS free, or what categories would you put them into?
Yeah, actually, I can say it is animal free because we source it from the gut of some mammalians, and we are done with the animals. So after sourcing it, we can continue our path by not intervening animals. So I can say it is 100% animal free.
Well, that's good context to know. And Jacqui, how would you address considerations for using iPSCs versus native animal stem cells for edible cell-based meat?
Okay, so I guess there's pros and cons to both. And I guess this question is mostly around the regulatory piece from the questioner. So I guess the challenges for iPS cells could be that obviously there might be some level of genetic modifications that would be involved to convert somatic cells back to self-renewing stem cell. And in our case, we use a non-integrative methodologies to convert our cells, which can then be subsequently cleared from the cells. We've spoken directly to the FDA and had feedback from a consultant on the EU FSA regarding the regulatory challenges for the use of these cells in the end product. So it's the end product that will be classified, and in the end product there won't be any genetically modified elements within the end product. So we are supporting our clients by developing a package for the cells, a safety package for the cells, as they'll be contained, and then they can put that into their dossier. And we don't see it as a key regulatory hurdle. And in fact, the FDA, I think, would be very open to that type of package for approval, as the end product won't contain any genetically modified elements because of the type of technologies we use to generate our cells. Our PGCs, for example, that we produce aren't genetically modified, so we're trying to, avoid those other challenges for some of the other host species that we're targeting in our cell line development.
Very nice, great. And Alex, on the flavor discussion, do you consider natural flavoring ingredients like those obtained from yeast cells, if they're available in very good chicken, beef, and fish flavors? Also, what do you think of plant-based meat alternatives?
I'll stick to segment. What do I think of plant-based alternatives? As an analytical chemist or as the consumer?
I think there's some room for interpretation there, but I'd encourage both answers.
Well, working with the company working on algae, that kind of convinced me, as a food additive with high fiber and other nutrient advantages, that, it seems like something that if it was part of a composition of a meal element or food product, I would probably choose it. If it was just kale sitting on my plate, probably not. But again, it gets back to, that there's a market for all this out there. The first question was about the taste elements. So you'd have to get back to the molecular level as to what taste elements are you talking about. Usually we're looking at fatty acids, amino acids, carotenoids, terpenes, phenolic compounds. And so, and taste at the end of the day has to be done by some type of taster, right? In the product, at the end product. So what we're really looking for, again, is just the compositional differences between product lines and between natural product and your cell-based product.
Great. And when you think about evaluating cell-based meat, a lot of these chemicals you're talking about, the place where I feel like I hear them most mentioned outside of the cell-based meat space is the brewing industry. So people who are home brewers might want to think about tuning this stuff. How does it compare?
Well, it's similar, I suppose. I mean, it is a food product, so it has to pass FDA inspection, of course, for safety, as you have a new brewery comes up with some type of blueberry beer or something like that. It is a fermentation process, so it's a culture grown product. So there's a lot of similarities between the two industries. Obviously, the products are different, and other elements, of course, than that. But in terms of just, I mean, I say this all the time, whether you're looking at onions or you're looking at yeast or you're looking at humans, we have the basic metabolism is the same. You need carbon, you need nitrogen, you need a glycolytic pathway to generate energy, you need catabolic and anabolic processes to generate biomass and to tear things down. So, we're always looking at very similar processes. The differences, of course, are what are the nutrients that are required, what are the cofactors that are required for a stable or prolific cell that's going to be a healthy growing cell. And that differs from cell type to cell type.
Great. That's really helpful context. For Kerem, what is the novel discovery process and techniques that Biftek uses to screen for and to find their key microbes?
Yeah, actually, I may not name the key techniques because I'm not a scientist which performs those experiments. But what I know, we pick some mammalians which we would like to produce, right? I mean, like cow, chicken, fish. So we use their byproducts, I mean, microbiomes in their guts, and like brute force attack, which we call it in computer science. We screen all of those microorganisms and pick some of the relevant ones for the cell growth part. And actually this comes out of 30 years of experience of our scientists. And now out of 180 promising bacteria and microorganisms, we scaled down to 5 now. And those 5 microorganisms are very beneficial in the cell growth. So I don't know the technical name, but we are picking the microbiome. Actually, this is called postbiotics. And those postbiotics have some beneficial relationship with the cell growth.
Great. Well, thank you, Kerem. One last audience question. Hi, Jacqui. Do you see a place for generating commercially available cell lines beyond iPSCs, such as myosatellite cell lines? And what are some of the limitations?
Cool, thanks. So if you mean by myosatellite cell lines, if it's adult muscle stem cells, then we haven't focused on that because they don't have the same self-renewing capacity that iPS cells would have, or embryonic stem cells would have, to assist us with the scale up. So we haven't focused on that particular area. We do consider different options for cell line development. So this is a space we'll continually look at, but because of our data for our iPS cell lines and their tremendous self-renewing capacity, we would see that as our main focus going forward, because I think that would be a key issue in scale up and quality control for adult stem cell lines in terms of the number of doublings that they can have, and then you would have to go back to the animals to derive more lines, and that has all sorts of associated disadvantages for your, obviously, your quality control and batch to batch variation. So I would say in the the midterm, our focus would be on induced pluripotent stem cells and PGCs, as I mentioned earlier. I hope that answers the question.
Great, wonderful. Well, so this has been a wonderful discussion. I've really enjoyed hearing from all of you. I think it's been really interesting to hear about how some of these cell line development technologies and analytical techniques could come together to help new companies and existing companies think a little bit more about what kind of tech they can bring to the table. So it's an open conversation as things go on. It seems like more and more tech gets integrated into the process, which is really exciting.